So today I'm going to be walking you guys through single-cell RNA sequencing. Basically what this is that it is a technique that collects data from genomes, epigenomes, transcriptomes, and proteins in individual cells for the purpose of gene expression, profiling them. Single-cell sequencing allows us to see gene expression in individual cells by scanning thousands at a time, making it a very efficient technique. Specific variations among cells and heterogeneous cell samples can be examined, as well as the quantity of gene expression. The general technique layout really varies. This is the pretty basic backbone of it. So you start with tissue preparation, isolating cells from environment. So this is done by mechanical isolation, enzymatic digestion, or combo both, depending on the cells. And this depends on types of cells that need to be prepared into a viable single-cell suspension because these cells do need to be in suspension for this to work. Second step is DCR. This is not always required, and we will get into that. If it is required, it is because cell viability did not exceed 70%. Moving on from that, single cells are isolated and compartmentalized. This is done by preparing the single-cell libraries using microfluidic devices, which we will get into, and then the libraries are loaded onto sequencing devices where further analysis is done by other technologies. DCR, dead cell removal, is magnetic beads are conjugated to antibodies that target an XNV, which is a protein that binds to phosphatidylserine. If you guys remember when we were talking about the plasma membrane, phosphatidylserine is only on that outer monolayer as a tag to show other cells that that cell is dead or went through apoptosis. So that is how those beads work as far as catching those dead cells. So to prepare the cell libraries, in particular, this is talking about the 10x Genomics Chromium Controller. There are other devices for this, but this is just more of a specific example. To prepare the cell libraries and separate out those individual cells, the cells are loaded onto a microfluidic chip where they're encapsulated by droplets containing barcoded gel beads and reagents for reverse transcription. So basically what's happening is after the cells are lysed, the beads capture the poly A tails of the mRNA, and then reverse transcription generates cDNA, which is the complementary DNA, where those cDNAs were previously tagged, mRNAs, and then after cDNAs were tagged with a barcode to identify the cell and where the cell came from, as well as a UMI, which is a unique molecular identifier to label the mRNA transcript. So these are important to the system because these are what help with that gene profiling down to the single cell level. The pooled cDNA is amplified in bulk, and then adapters are usually added to flank the region to be sequenced, so that just helps with the cDNA amplification. And the sequencing libraries are just collections of DNA fragments with adapters attached to them. So that's what I mean by cell libraries or sequencing libraries. And then following that, the libraries are loaded onto sequencer devices. In this figure, it's showing an Illumina sequence device. There are other ones as well. I think these aren't as diverse. I'm pretty sure these are pretty similar across the board. But in specific, this was an Illumina device used for analyzing. Not analyzing, but just taking in the data and recording it. Then data analysis and visualization can be done by certain software. This is called Bioinformatics Pipeline. It's actually pretty cool if you wanted to look it up. It's called NGS Bioinformatics Pipelines, which just stands for Next Generation Sequencing. And it's just a collection of processing elements and algorithms that help with analyzing the data from the sequencing and really showing those individual gene expression profiles, which seems pretty cool. So that's about it for this. Yeah, thanks, guys.